Introduction to Medical Virology. So now we are moving to another section, that is virology. As you know, I think about the smallpox, variola virus. It was arise in 10th century BC. There are records in epidemics in Asia. It's a highly infectious disease, often fatal. Variola forms spot, vesicular rash. Chinese prevent infection by self-inoculation of pus lesions. It is termed variolation. Later on spread to China, England, similar infection in cows and milkmaids infected by cowpox do not get smallpox. So it implies kind of immunity. Use the pox from cowpox lesion to inoculate and prevent smallpox, term vaccination, vaccine as cow. Later on in the 20th century, use of chick embryo, tissue cell culture to study virus in the lab. Look, evolution. Research and development for biological warfare, USA and Soviet Union, used bad use. Later, in World Health Organization begins program to vaccinate susceptible persons, 1977. Last natural case of disease in the Bangladesh, 79. WHO declares smallpox was eradicated. It's one of great achievement of mankind against a viral disease. Nowadays, COVID-19. Imagine about the burden pandemic nature. Likewise, viruses are one of the key agent to cause infections leading to burden in mankind. What are viruses? They are non-living. They have no cell nucleus, organelles, or even a cytoplasm. Viruses show property of living things only inside the living cell. They are pure parasites, one way. Outside the cell, they behave like a non-living thing. They are called obligate intracellular parasites. Virus particles contains only one kind of nucleic acid, either DNA or RNA, but never both, because they can't reproduce by themselves. They can't reproduce themselves. They need the host. Viruses are not considered living. They are obligate intracellular parasites. They possess DNA or RNA by the means of their messenger system. They They utilize host compound to make their next progeny. They are very small, much smaller than the cells of living things, and are basically just package of nucleic acid and proteins. So least adaptation. One is a maximum adaptation for the survival. There are different sizes. So tiny infectious particles that can reproduce only by infecting host. So virus command the host cells and use its resource to make more viruses, basically programming it to become a virus factory. Structure of the virus. You can see envelope. Not all possess envelope. Some are non-envelope. In a capsid and nucleic acid genome is there. Protein, protective protein shell is termed capsid. Nucleic acid genome and layer of the membrane called envelope. Virus capsids. Capsid or protein shell. The virus is made up of many protein molecules. The proteins join to make this unit called capsomeres, capsomer, which together make up to the capsid, just like football. Capsid proteins are always encoded by the virus gene, meaning that it's the virus, not from the host. Capsidomes of virus are relatively simple and are made up of multiple copies of a single protein. Capsid protein, 5 copies, capsid proteins, 1 capsomere, and 12 capsomere capsid. It depends. Okay. So capsid comes in many forms. Isocohedral capsid at 12 phases, multiple phases, and named after the 20 side shape called isocohedral. Filamentous, just. They are linear, thin thread-like appearance, head tail. Capsid are kind of hybrid between filamentous and isocovertral. Contain filamentous head and isocovertral head. Envelopes. In addition to capsids, some viruses have lipid membrane. It is termed envelope. Virus envelopes can be external, surrounding the entire capsule, or internal, found beneath the capsid. It's a typical bilayer virus with only a capsid and no envelope are known as non-envelope. And their projections referred to as spikes may or may not external from the viral envelope. These surface projections are termed glycoproteins, made up of glycoproteins, are served to attach virons to specific receptor sites on host cell surfaces. So there are hemagglutinins, so property useful in viral identification, hemagglutination inhibition test, or other way, hemagglutinate, which acts on several cells, red cells, and form clumps, hemagglutination. About the virus genome, the genetic material I said contain either DNA or RNA. DNA can be double-stranded or single-stranded. RNA also same. Viral genome also comes in various shapes, sizes, and varieties. They typically range from 2,000 to several 100,000 nucleotides in length. What is the viral infection? Viral infection means these viruses are using your cells or host cells to make copies for themselves. The viral life cycle is set steps. Virus recognize and enter the host cell, and later they reprogram the host by providing instruction to form the viral DNA or RNA and use the host resource to make more virus particles. For that, there should be attachment. Virus recognize and binds to host cell via the receptor molecule on the surface. So infection cycle begins. Mode of route of entry, now it's attachment. Then it should enter to the cell. Then genome replication and gene expression. Then they form assembly. New viral particles are assembled from the genome copied in the viral protein. Then they are released. This cycle may be disruption of lytic or lysogenic like, so they come and infect to release and they go for infect another cycles and cells, and the cycle continues. About the attachment, bit more. Specific protein on the capsid of the virus physically sticks to the specific molecules on the membrane of the host cells. Receptor, messenger, it's a binding receptor, and they enter. The molecule called receptor is usually protein. Virus recognize its host cell based on the receptor they carry. So there is a tissue tropism. Tissue tropism means specific virus goes and acts on either one group of cell or multiple cells where they have in the receptor more. Imagine respiratory virus. That's why we say tissue tropism. They are more favored to adhere to the respiratory epithelium. Some may gastric epithelium, some may to platelets, some may to vasculature. Likewise, endothelial cells, that's their tropism is there. Once they attach, they tend to enter. One typical route for viral entry is fusion with the membrane, which is most common in the viruses with envelope viruses. But non-envelope virus, they take in them as a bulk transport called endocytosis. So then they goes for the replication of genomes. Involves copying the viral genome and making the viral proteins so that the new virus particles are assembled. The material for the process comes from the host cell, not the virus. So pure parasitism. Most of the machinery for the replication and gene expression also provided by the host cell. So they may have in the virus may have a few enzymes, few messenger molecules, and they initiate the replication of DNA or RNA. The messenger RNA encodes viral genes are translated into the viral protein using host cell ribosomes. All the things are utilized from the host. However, certain mistakes, copying of RNA virus genome cannot be performed by the host cell enzymes. In such cases, virus must encode their own enzymes. Some instances, in few steps, they possess their enzymes. So viral genome, gene expression and replication copy of the genome. All virus must encode capsid protein, and envelope viruses typically also encode envelope proteins if envelope is there. Then they go for the assembly. Newly synthesized capsid proteins come together to form capsomere. Now they form the capsomere, which interact with other capsomes to form the full size capsid. Some like head tail viruses. First assembly of empty capsid, then stuffed with the viral genome inside. So different mechanisms are there. Release. Last step in the virus life cycle. Different types of viral exit different modes. Some may also process by lysis, some by the endocytosis without lysis. Lysis, exocytosis, sorry, and maybe budding. So this is the life cycle: attachment, entry, replication, assembly, release. Bit of virion. A virion is a structurally complete virus that capable of infecting new virus or new cells. Sorry. So that is usually imagine attachment, assembly, and release. Now it is a virion, structurally complete virus that capable of infecting new cells. Bit about the classification of virus. Phenotype characteristics such as morphology, nucleic acid type, mode of replication, host organisms, and type of disease they cause, depending on those factors we classify. International Committee on Toxinovirus, ICT, or Baltimore Classification System. Just you don't want to get any much more idea of this. Think there is a get idea there are systems of classifications. Similar to bacteria, order, family, subfamily, genus, so far there are classifications. Herpesiridae, you want to know, Echinovirus, likewise. About the Baltimore, 1971 years, DNA or RNA strand, single or double sense, positive sense, negative sense, and method of replication. There are grouping, but it's not correct. So double stranded, you have to know. It's a pathogenic virus, adeno, double stranded DNA viruses, single stranded DNA viruses, double stranded adenoherpes pox, single stranded PAO B19, like double stranded RNA virus, reoviruses are rota. Single stranded RNA viruses like positive sense, Picano, Toga virus, rabies, and single stranded RNA viruses, negative sense, Orthomyxin, and rhabdovirus. Likewise, retroviruses, that is HIV virus, and hepatinoviruses, which cause hepatitis B. So double stranded DNA virus. So Baltimore classification there, but what I said earlier, it's the most simple. Just get again idea. So there are other one, bacteriophage like. So it's for the class sake of completion I have put, but it's not. Bit of emerging infections. What is the definition of emerging infection? It's time to read you, because now also we are in a pandemic. It's a kind of emerging infection. So it has newly developed a disease. So that's term emergence. Or maybe it's earlier we term as not an infection, but later on it's identified as an infectious disease like gastritis, Lyme disease, like gastritis following Helicobacter. Early it was thought it's not an infectious infection. So it's also emerging infection. Or less, it was term further re-emerging. Early it was there, then subsided, but later on it's emerged as a massive scale. So year of emergence and site, geographical locales there. Plague, SARS, West Nile virus, Legionellosis, tularemia, cholera, polio, Ebola, likewise. A bit of Ebola virus, fatal hemorrhagic fever, HIV, chronic infection, immunodeficiency, hantavirus, I think you heard. There is a renal CKD etiology also they are going on prion, and severe acute respiratory syndrome, avian influenza and COVID-19. So when you know the replication cycle of the particular virus, we can target antivirals. So antivirals mainly broadly classify targeting viral function or targeting cellular functions that virus needs. Targeting viral function, they act on nucleic acid polymerases, which required for replication, proteases, integrases, neuraminases. So based on that, we do. So we will later specifically learn about much more about these antivirals. Thank you.